Okay, so a pleasant good afternoon to everyone. And today we'll be looking at vaccines and the immune system. Since it is that we find ourselves in the era of COVID, I did think it was pertinent and relevant at the same time to have a look at it in the context of the WHO's mandates relating to COVID. So this presentation for today comes from the WHO, in which we will incorporate those things relevant for our class as it relates to immunization and vaccination. We'll relate those things to the COVID mandate as issued by the WHO. So let's have a go and begin. All right, so while we get to the relevant slide, this is the first slide. When we look at the global situation surrounding COVID as of February the 2nd, 2022, in terms of the number of cases, it has been in excess of 380 million, with deaths approximately 5.6. If we were to look at the graph here, we would see that there was a peak somewhere between November and April of 2021, 2022. Going forward there, we did see there was a bit of a spike in the number of cases. And this would have been 2020 to 2021, because this year represents 2022. So this would have been November 2020 to April 2021. We did see a bit of a spike. And of course, vaccination did come into play, and we saw a decrease in the number of deaths. Interestingly, in terms of the regions in which we do have the most number of cases, highlighted here in the green, we do notice that the European region, they account for the majority of the number of deaths, and they are seconded only by regions of the Americas. Interestingly enough, in the regions, the African regions, they're virtually negligible in terms of the number of deaths in the wider scope of things. So therefore, the African region has virtually the lowest, not virtually, but it does have the lowest number of deaths, whereas the European region, seconded by America, account for the majority of the deaths. So in terms of the vaccines themselves, the vaccines are delivered on different types of platforms. So there are different types of vaccines, but ultimately what the vaccines do is help prevent us getting ill. Or if we do get ill, we experience a slighter version and not the full-blown version of the disease itself. And in some cases, of course, the full-blown version, as we're well aware, is lethal. So it does prevent deaths in that regards. Different platforms in terms of mechanisms, delivery methods, and also the processes through which these vaccines work. We'll be looking at them in a little more detail very shortly. Some of the vaccine technologies, such as the mRNA vaccines related to the messenger RNA, have been licensed for use for the first time. One of the things which has come out of COVID is the use of these mRNA vaccines. And going forward, not only for COVID, but for other diseases, we will be seeing the rise of these mRNA vaccines. It has thus far been proven to be relatively successful in terms of both application, delivery, and outcomes. And one of the good things about these, it's easy to replicate and distribute. So therefore, do look forward to greater things from the mRNA vaccines. As of February of this year, there were 8 different types of vaccines. And again, we'll look at those shortly. The current vaccines, they continue to provide strong prevention or protection against severe death. However, some are less effective against one of those variants, the Omicron. Fortunately for us, the Omicron has not manifested itself to any great deal in terms of showing an increase in the number of deaths. So thus far, the Omicron variant, granted it is a more lethal type of variant, but the manifestation in terms of the number of deaths globally, that has not come to the fore. So in terms of the immune response to a viral infection, so what happens in terms of it? Well, there are two types of immunities, the innate immunity and the adaptive. Innate, a general type of immunity, which is a response to any infection. And this is a naturally occurring immunity, and it generates a non-specific immune response. This one is always, in terms of the innate, is always present in the body. And in terms of its speed of action, it is a very rapid response. So its components include the plasma proteins, phagocytes, physical and chemical barriers. These form the components of the innate system. Very importantly to remember, with the innate system, it does not develop memory cells. The scope of its diversity is indeed smaller. It is less potent than the adaptive immunity. It does not produce allergic reactions such as redness and swelling. So when we do look at the adaptive, it's very specific, involves both T and B cells, and this generates in response to exposure to an external factor. One of the things which we will notice as it relates to the adaptive immunity, it's delayed. So it usually, in terms of this cellular response, from 5 to 6 days before when it actually kicks in to do its work. Now, in terms of the humoral and cell-mediated immunity, these are the types that form the components of this system. The lymph nodes, spleen, and lymphoid tissues are the barriers, which is oftentimes why when you do go to the doctor, you will see him or her palpating your lymph nodes. This is one way in which an infection is actually checked. The adaptive immunity also develops memory cells. It does possess a higher degree of diversity, and it does exhibit a higher potency. All right, so these are some of the salient points relating to both innate and the adaptive immunity. Let's talk a little more detail in them. So the innate immune response, when a virus enters the body, the cells can recognize various markers, and these markers, most of the times, they're proteins present on the membrane, the cell membrane itself. Could be receptors, could be unique protein presence on the membrane themselves. This results in a non-specific antiviral activity, and the cells of the innate system are activated to remove these pathogens and foreign cells. So if it is that a pathogen or a disease-causing cell presents itself to the body, in terms of the innate immune response, you will get this response when it does recognize a non-self. One of the major properties of the immune system is that ability to recognize self from non-self. When non-self is recognized, you have activation of macrophages, neutrophils, natural killer cells, or dendritic cells, and they, of course, respectively have phagocytotic activity. The neutrophils, in terms of release toxins that could kill, perforins, for example, that permeate these membranes of these pathogens, making them porous, and this could subsequently lead to activation of apoptotic pathways, which will then degrade the material of the cell via the release of lytic enzymes from the lysosomes. You have natural killer cells, which recognizes specific antigens, which are actually presented on the cell membrane, and then the cells can then latch on and kill these cells that are actually associated with these pathogenic proteins. Then, of course, you have dendritic cells. Again, these are antigen-presenting cells, and this, of course, once the antigens are presented, they could then trigger an adaptive immune response. So let's have a look at that adaptive immune response. Here we have a virus and a white blood cell in terms of recognizing the virus, phagocytotic activity, and then we have a viral antigen is presented on the membrane of the phagocytotic cell itself. The helper T cell then recognizes the specific fragment from the virus, as shown in this case, and then it signals B cells. B cells then produce antibodies, which themselves could then bind to specific receptors on the virus. Let's say it's MHC class 1 or 2 type receptors present on the virus, which are very important for attachment to the cells prior to infection. So these antibodies can then bind, so that will then prevent binding of the viral particle or cell to the healthy cell. Remember, viruses have to get into the healthy cell, hijack its machinery, and that's how they replicate. So therefore, if the antibodies bind then these receptors, they will not be able to bind to the healthy cell. If they don't bind to the healthy cell, well, they can't replicate. And that is one way of keeping viral load in check. Another way of the T cell, they signal cytotoxic T cells, which recognize the specific antigen. And as we mentioned before, they could then, when they bind to the virus, they could release perforins, which then cause the membrane to become porous and then induce apoptotic mechanisms within the cell itself. So let's talk about the adaptive immune response from the perspective of the B cells. The B cells, which are mature in the bone marrow, they produce antibodies that are very specific to the virus. The IgM antibodies are produced first, and then they are followed by the IgG. These are produced at the same time, and they could remain within the milieu of the cell, could remain within the organism itself, and they could remain there for months or even years. Memory cells, very importantly, they could also be produced. And once infection is over, these cells, even though they decline in number, they are still present, such that should a reinfection occur, the response of the body's immune system would indeed be very fast. So this is showing the response in an immunized person to an infection. And here we see, in terms of the cellular response, the virus amount, note the virus amount actually goes down, and the antibody response. Note as the antibody response maximizes, we see a decrease in terms of the viral amount, which shows the efficacy of the antibody response. So let's talk about the immune response, which could be induced by vaccines. So what vaccines are really doing is they're just mimicking a viral response, as shown if we're looking, in this case, we're looking at COVID. So it's mimicking it, and it's getting the body ready such that should it be infected with the virus itself, it could now be prepared to launch an attack on it. So let's have a look. So the CD4, the vaccines, they do deliver an immunogen, an antigen which is able to elicit an immune response to the immune system. The CD4 plus helper T cells, these are CD4 positive, which means they show the receptor on the surface. The CD4 helper T cells, in turn, they stimulate B cells, as was shown previously. If you just a step back, right? Here we see activation of B and cytotoxic T cells. So the B cell to produce neutralizing antibodies, as we mentioned, they could bind then to the receptors, adhesion molecules, for which are very essential for the virus to bind to the healthy cell. So if the antibodies are binding to these receptors, as mentioned before, the virus will now be unable to latch on to the healthy cell, will not be able to get its viral material into the cell, will not be able to replicate, and by extension will then die, or will not be able to replicate. The CD8 positive cytotoxic T cells, they recognize and kill infected cells infected by the virus, as mentioned previously. And again, this is done by 1, perforins, which actually penetrate the membrane itself, and 2, by activation of the apoptotic pathway. So in terms of the currently existing vaccines, which mechanisms do they employ? Well, there are four major vaccine platforms, namely mRNA, recombinant, inactivated virus, and the recombinant spike protein nanoparticles. The types which use the mRNA are the Pfizer and Moderna vaccines, the recombinant, Vaxzevria, Covishield, and Janssen, and the inactivated would be CoronaVac, Sinopharm, and Covaxin. These are the inactivated type. This, of course, is the more traditional of the vaccines in terms of an inactivated virus being used, inactivated in terms, which means the virus would not progress in terms of causing the disease itself, but it would stimulate the immune system to give a response, and recombinant spike protein. So in terms of their description, the mRNA, this vaccine provides the instructions to make part of the spike protein, and this triggers the recipient's immune system to develop a protective response, which defends against future exposure to SARS-CoV-2. Right? So mRNA, and as I mentioned, this is one of the mechanisms which will be employed for the future. So with monkeypox on the horizons, fingers crossed, hopefully it will not become a pandemic. mRNA would more than likely be the vaccine methodology of choice in terms of rapidly producing vaccines. The recombinant viral, it's a modified virus other than the virus causing the COVID, and is used to deliver the instructions to the human cell to make part of the SARS-CoV-2 spike protein. And this protein triggers the immune system to develop a protective response. So you have to remember antigen-presenting cells, right? When they do show the protein, it stimulates the body's immune system now to either activate B cells or the cytotoxic killer cells, killer T cells, and in so doing will limit the viral spread and the viral load within the body. The inactivated Sinovac, Sinopharm, Chinese vaccines, and this is an inactivated vaccine, consists of killed virus particles that are recognized. So even though the virus particle is killed, meaning the DNA component is no longer lethal, they are the protein, the proteinaceous shell is still there in terms of the membrane, and these can be recognized and cause the immune system to launch its domino effect as it relates to pathogenic protection. Then you have the recombinant spike protein. Subunit vaccines contain specific fragments of the SARS-CoV-2 spike proteins. The spike proteins, these are on the, if we were just to go back, these are the spike proteins here, right? So these are the ones which are present, and these are the ones that are recognized by the immune system in terms of giving the response. Let's see if we could look at each one of these methods in a little more detail. So the mRNA vaccines, right? mRNA stands for messenger RNA. We have to remember in terms of the central dogma of biology, we have DNA is transcribed into RNA and then translated into proteins, right? So the mRNA are antigen coding strands delivered inside a coat. So the message is actually being placed into the cell itself. Once inside, it's translated into the protein, and the antigen is recognized, inducing, right? So it's recognized and it induced an immune reaction. And this immune reaction is very similar to the one as if a virus is actually within the cell itself. So this mRNA-derived protein is recognized as a pathogen, and it stimulates a strong response. Some of these vaccines include the Pfizer and the Moderna type vaccines. So here it is in terms of the antigen-presenting cell, and the T helper cell causes either 1, cytotoxic T cell activation, or 2, B cell activation, which will then lead to antibodies being produced. The viral vector vaccines, these use a non-coronavirus vector modified to include a gene that encodes a target antigen. So again, the gene is encoded, right? Very important. ultimately what happens is that you do have, in this case, protein being produced. This looks very much like the spike protein, which is then presented, recognized by the T helper cell, which could then cause either B cell activation to produce antibodies or activation of cytotoxic T cells, which would be able to recognize cells which have this particular spike protein and then kill them, as mentioned before, either by inducing apoptotic mechanism or by causing perforin to actually lyse the membranes, thereby destroying the contents of the cell itself. So some vaccines which are of this type include the AstraZeneca and the AstraZeneca vaccine, which is also produced by the Serum Institute of India, and the Janssen vaccine as well. It's very important to know that all WHO viral vector vaccines, they are non-replicating viral vector vaccines. Then we have the inactivated virus. So in this case, the genetic material has been destroyed. So therefore, there is no hope of replication occurring. However, you do have some of the proteins which are present, which could be recognized by the immune system that identifies it as a viral pathogen. So therefore, the use of this inactivated viral vaccine, it then stimulates the body to go through its steps as if it is recognizing viral invader. So the inactivated virus, it cannot replicate because the genetic material has been destroyed. And sometimes an adjuvant is used, molecules that stimulate the immune system. And this is used to strengthen the immune response. And that is very important in terms of these adjuvants, right? So, and you might be saying then, okay, so these adjuvants, what specifically are they? Well, there are some ingredients which are, added to vaccine, which help give a stronger immune response. All right, so those are very important in terms of ensuring that the response that the body gives is maximal as it relates to the use of that particular vaccine. And then we have the viral subunit vaccines, and they use the antigen of the virus without any genetic material, right? And again, an adjuvant will be used to give a better immune response. All right, so as shown here in this diagram, right? So here it is the adjuvants, and some of the adjuvants which are used include aluminum, sometimes compounds of aluminum, such as the hydroxyphosphate sulfate, aluminum hydroxide as well. Sometimes, which is why it is important, should you have or known for reactions to some of these adjuvants, you should be careful in terms of which vaccine you take to identify the adjuvants to ensure that they do not do any harm to you. If it's known you have specific responses to these adjuvants. So in terms of these subunit, with the help of antigen-presenting cells, they're recognized, and the vaccines generally induce antibody-mediated immunity. All right, so those are the 4 types which we mentioned in terms of those vaccines. And notwithstanding that, it's very important that the WHO emergency use listing involves careful and rigorous assessment. So once you hear, or once it is given, the WHO seal of approval, do know it goes through a very rigorous system of quality, safety, and efficacy data. And after going through these by peer review, with using experts from the field itself, is only at this point would the WHO give a preparedness phase, would give would give the assurance that it could be used. Now, some of these, in terms of the methodology, there's a preparedness phase in which you establish an assessment platform which assesses the vaccine. There's the emergency phase in which conducts rigorous assessment and includes a recommendation is issued, and there's the post-listing phase in which continuous collection of data is occurring and actively monitors it for side effects. Interestingly enough, when we were looking at the COVID vaccine, we did see in certain instances, because of the fact that you had to weigh two very important parameters. One, the need for a vaccine against two, increasing deaths. So in certain instances, there was granted that there was oversight being done, but usually, so the time frame which would have taken on the order of years, this was now compressed onto the order of months, because of the fact that, yes, it needed to get this vaccine out there, because indeed we were in the throes of a pandemic. So this was one of the unique instances in which the rigorous testing was compressed into a very short space of time. Notwithstanding all of those things, do remember, continuous, while it is that now we see relaxation of transport to the US. The US has now relaxed its issue as it relates to testing in terms of travel to the US, but we still have to be careful. And in terms of protecting yourself, do remember to use all of these 6 methods which have been outlined by the WHO: distancing and keeping, washing hands, and limiting the spread of the virus. And most critically, of course, after you have examined all of the data concerning the virus itself, you should think seriously about getting vaccinated. All right, and that is the end of the presentation for today. Thank you very much. Do have a good one.